Hello, can somebody confirm that you can hear me?
Good.
Great, thank you. All right, I keep having trouble logging in. Today it said your session has expired and you can no longer log in or something like that. So I had to refresh the page. I don't know what's going on with my ability to log in here. All right, let me share my screen. I guess we can close that, but I can't reach it at the moment. Maybe now I can. It was just this page I had to refresh. I don't know why that's the case. All right. So today is February 21st, and we're supposed to talk about Lab 9, identification of gram-negative organisms. Please recall that you're also supposed to be working on your unknown project, and you only have about a week further to work on your unknown project. So those of you who haven't started, you're probably not going to have time. You'd have to be extremely lucky to finish in 1 week. If you're confused, like I said, on what to do, just contact me and I'll try and help you. All
I
right.
have a question about the unknown project. I received a test and I've been keeping track of them like we're supposed to. However, on the chart, my results don't align.
Okay, I would like to talk to you privately about this because this is something unusual and I don't think the other students will get much information out of here. If the chart isn't agreeing with what the results I sent you, my guess is you're interpreting the results incorrectly because the chart should be correct.
Okay, is it okay if I stay afterwards and we can talk
Yeah, we can
about it
talk
in
about it
private?
after the lab or after I'm done talking in the lab, either way. Yeah,
Okay.
okay. All right, because like I said, I don't think that'll be terribly useful for most of the students. All right, let's talk about Lab 9. Are there any other questions before we get started, I guess? All right. Scroll up to the top to remind me I'll be here at least until 8 o'clock, maybe as late as 8:20. Generally, students, you guys have been logging off early, and that's not a problem. But when the last student logs off, I'm going to log off as well. So if you have questions, make sure you log in to ask them early. Let's see. I've got it up here, just looking to find it. Here it is. Lab 9, identification of gram-negative bacilli. We have some case studies today and of course the Lab 9 worksheet. There are some reading to do, not too many pages to do before you begin this lab. Some learning objectives. Upon successful completion of this lab, you should be able to interpret the results for the following biochemical tests. The IMVIC reactions, and this is actually 4 different tests. The indole test, that's what the I stands for. The methyl red test, that's what the M stands for. This is the lecture file just finished. Voges Proscure or VP, and I'm not Russian, so I could be butchering that name, but that's what the V stands for. The I is just a filler, and then the C for citrate test. We're also going to talk about the carbohydrate fermentation test today, the motility test, and the test to see if your organism produces hydrogen sulfide. You should be able to explain how to interpret the triple sugar iron slant, that's the TSI, the Sims deep test, and then growth on McConkey's agar, as well as growth on HE or hectone enteric agar. All of these tests are running more than one test at a time for the triple sugar iron and the Sims Deep. I believe they're running 3 tests at one time. McConkey's, I think, is, running 2 tests at one time. And then lastly, use biochemical analysis to solve case studies involving gram-negative bacterial infections. So members of the family Enterobacteriaceae, which are called enterics for short, are small gram-negative, non-spore-forming bacilli. They are facultative anaerobes, and they're essentially everywhere. Enterobacteriaceae has 53 genera, so in a sense, it's one of the largest, if not the largest family of bacteria. And there are 24 genera that have been known to cause human infections. They are all glucose fermenting, oxidase negative organisms. Most of these genera are normal microbiota of the intestinal tract of animals, so we call them AKA coliforms. However, some of them are normal in some animals, but not in humans. For example, Salmonella. And what's another one that's normal in another animal? I don't remember if it's Edwardsella or Shigella, are normal in like birds, their intestines, but they're not normal in human gut or intestines. And if you have them in your gut, you'll get sick. They are all opportunistic pathogens in that they can cause urinary tract and wound infections, pneumonia, and septicemia. Common genera responsible for causing opportunistic infections include Escherichia, like E. coli, Enterobacter, Klebsiella, Citrobacter, and Serratia. Several of the genera cause gastroenteritis. These include organisms in the genera Salmonella, Shigella, and then E. coli, Serovar 0157H7. Normally, E. coli is in our guts, and it is a normal part of our microbiota, and it does us actually good. It helps us digest our food. However, E. coli, Serovar 0157H7 is a human pathogen, and it usually causes disease. Pseudomonas aeruginosa is in a similar family to the Enterobacteriaceae. It's the Pseudomonaceae, and these organisms are similar to the Enterobacteriaceae in that they are gram-negative bacilli. However, the Pseudomonas are non-glucose fermenting, oxidase-positive bacteria. They can cause opportunistic pathogens causing infections, particularly in the immunosuppressed individuals, burn patients, and trauma patients. Pseudomonas aeruginosa is a human pathogen, and it is a health-acquired infection. So healthcare-acquired infection. And we will call that later a nosocomial infection. But in this terminology, it's a healthcare-acquired infection. Meaning you can find it in most hospital environments. It lives wherever there's moisture. It's even in dilute soap solutions, can grow there, in flower vases, sinks, drains, other things. So it's very common. It's important for microbiologists to be able to identify and perform antibiotic susceptibility testing on gram-negative bacilli from cultures obtained from clinical specimens. In this lab, we're going to talk about the common biochemical tests that are discussed to identify gram-negative bacilli. And here are some of the tests here. We won't use these, but you could run many tests if you injected this with your sample you're testing. But we're going to talk about the indole test, which is actually 4 here. Some organisms produce the enzyme tryptophanase that converts the amino acid tryptophan into the products of indole and pyruvic acid. You just grow the bacteria in something that has the amino acid tryptophan, and it could just be protein which has the amino acid tryptophan. After overnight incubation, you add Kovacs reagent. Kovacs reagent will react with indole to produce a red color. E. coli tests positive for indole production. And so we would see this here, where this is the added Kovacs, the yellow, and this is supposed to be reddish. It's kind of a purplish here, and that's the photograph. Sorry about that. It's the photograph we have and the photograph that is used in this lab. But anyways, if it turns reddish, it's positive for the production of indole, in which case tryptophan was converted by the enzyme tryptophanase into indole and pyruvic acid. If it's negative, indole is not produced, and it'll be a yellowish color. The Kovacs reagent will remain yellow. Any questions about the indole test? All right. The methyl red and the Voges Proskure tests are 2 tests, but they're usually started together in one tube, and then that tube is divided into 2, and half the tube we use for the methyl red test, the other half we use for the Voges Proskure, and I'm just going to call that VP test. Okay. The MR test and the VP test are used to detect what type of end products are formed from glycolysis. Methyl red looks at acid products being produced, and VP looks at neutral products being produced. The methyl red test detects acid products, and VP test detects neutral products. So you have a MRVP tube that has a broth in it, and that broth contains peptones and glucose. You then inoculate that with a bacteria and grow it for 2 to 5 days. Let me see if I can remember this right. Methyl red, it's best if you grow it for 5 days, and VP, it's better if you grow it for 2 days, although you could grow it for 5 days. Methyl red is added to an aliquoted portion of the broth. If the bacteria produce acid products, the pH indicator remains red, and this indicates positive test result, meaning that acids were produced. The other half of the tube you use for the VP test, you have to add Barrett's solution A and Barrett's solution B, and you shake it to add oxygen. You let it aliquot and develop, and after about 60 minutes, look at it. And if there's red, particularly in the top of the VP, then acetone, a neutral product, was produced. If it's not red, meaning it's all yellow, then it's a VP negative. Acetone was not produced. I guess I should have showed you the MR test. You add the methyl red to it, and if it's red, then that's MR positive. If the indicator turns to yellow, then it's MR negative, meaning methyl red, when you add it to the tube, it's usually red, but if it is not acidic, it will lose its color red, and change to yellow. And that usually happens immediately. All right, the last of the IMVIC tests is the citrate utilization test. This test tests the ability of the bacteria to use citrate as its sole carbon source. So in the citrate tubes, we only have one carbon source, and it is the sugar citrate. If the organism can grow in the tube, it will change the color of the tube to blue from utilizing the citrate. And this is citrate positive if it's blue. If the organism does not grow in the tube or it does not use the citrate, then the tube will remain green in color. What's happening is we have a pH indicator, which is bromothymol blue in the tube, and at alkaline, or is it acidic? Let me think about that. It's alkaline. At alkaline pH, it's blue in color, and non-alkaline, it remains green in color. Citrate, if it's used, produces, well, the nitrogen will be metabolized, and the nitrogen is not in the citrate, it's in either the ammonia or the urea. Is that right? It's not urea, because that would be a carbon source. It's a nitrogen in there. And anyways, it'll produce a basic solution if the bacteria uses the citrate and it grows. And so that would be citrate positive, citrate negative. For those of you who are interested, you don't need to know this for this lab, but citrate is in the Krebs cycle, and it's actually the first sugar after oxaloacetate combines with acetyl coenzyme A to make citrate. All right. That's the citrate test. Any questions about the citrate test? If not, let's talk about the carbohydrate metabolism test. Carbohydrate fermentation test is used to determine whether bacteria have the ability to hydrolyze a particular sugar. We put one sugar in the tube and then test the bacteria to see if it can ferment the sugar. We have several different sugars we can use, and frequently we're using glucose, fructose, lactose, sucrose, maltose. Actually, maltose we don't use. Mannitol. I think we use inulin also in our lab. And if the bacteria can ferment the sugar, it'll produce acids, which will change the media color to yellow. If the bacteria ferment the sugar and produce gas, you'll see that the color of the tube is yellow, and we'll have a bubble produced in the inverted Durham tube in the carbohydrate fermentation test. This is the inverted Durham tube right there. If it's negative, the media won't change color, or it'll become more pinkish, and that's negative for the carbohydrate fermentation. This is positive. You mark that as A for acid, and then this is marked as AG for acid and gas. Any questions about the carbohydrate fermentation test? Remember, the carbohydrate fermentation test is the glucose fermentation test, the sucrose fermentation test, the lactose fermentation test, and any other sugar that you are testing. And you're testing one sugar at a time. All right. If there's no questions, let's move on from the carbohydrate metabolism test to the motility test. Some bacteria possess flagella, and they are motile, they can swim through the bacteria. Other bacteria lack flagella and they are non-motile. In the motility test, we have a semi-solid agar media that stab inoculated with a bacteria and then incubated overnight. If the organism is non-motile, the bacteria will only grow in the stab, which you can see here. If, on the other hand, the bacteria have flagella, it can swim through this semi-solid media, and then you'll either see feathering along the stab mark, which is growth spreading out, or if the growth, it's very motile, you'll see growth everywhere, in which case the tube will appear cloudy. Let me see if I can show this better, the cloudiness. This tube, you can sort of see it's clear right there and right there. This tube is not, it's cloudy. And the cloudiness is from the bacterial growth. And another way you can tell from the cloudiness is you can't see the stab mark, but it would be right down here. Any questions about the motility test? Oh, I forgot to mention that the motility media has a very low agar percentage, and that's why we call it semi-solid, and the bacteria can swim through the agar of the motility test. Any questions about any of that? So this would be non-motile, that's motile. And the motile doesn't have to be like this. Like I said, it could be, this looks feathered, where there'd be growth like that. All right, hydrogen sulfide production is looking if the bacteria can produce hydrogen sulfide, a gas. We can test this in at least 3 different ways. You can test it in the triple sugar iron agar slant, and that's called the TSI slant. The sulfide endomotility agar deep, and that's usually called the SIM deep test. And then you can test it on the HE or the hectone enteric agar plate. In all 3 of the tests, if the bacteria produce hydrogen sulfide, the tube or media will turn blackish. And that's from hydrogen sulfide interacting with the indicator, forming a black precipitate, which is shown, excuse me, right here. Usually you see the black on the lower portions of the tube because you get hydrogen sulfide being produced anaerobically. When everything is growing aerobically, you will not have hydrogen sulfide production. So on the top of the tube, the bacteria may be growing aerobically and it won't be black on the top. This is very little, but a very little bit of the top is not black. And the lower portion of the tube is growing anaerobically and it will be black. In this case, most of the tube is black, but it doesn't have to be most of the tube. How you inoculate the hydrogen sulfide production tube is you just have a needle that's shown here, and then you stab inoculate the SIM deep. This is negative for hydrogen sulfide production, the one on the left, and the one on the right is positive for hydrogen sulfide production in a SIM deep tube. You can also test the hydrogen sulfide production in a TSI or triple sugar iron tube. In the triple sugar iron tube, you use a needle and then you take the needle. Let's see if I can get something close to a needle. And what you do is you roll the needle. So this would be the needle. You roll the needle in the bacteria, picking it up on the tip. And then you inoculate the bacteria at least 2/3 of the way down. So that should actually go down a little further because you want it to grow anaerobically. As like I said, hydrogen sulfide production is usually made anaerobically. If it may only be made anaerobically. And then you take the needle out and then put fresh bacteria on the needle and then inoculate the slant in a serpentine motion. What we're doing is we're growing the bacteria anaerobically right here and aerobically on the slant. I don't think I have a picture of TSI that's anyways, if it's black in the lower portion of the tube, then hydrogen sulfide is produced. I should mention that both of these tubes are looking at more than hydrogen sulfide. At the moment, we're only looking at hydrogen sulfide production. You can also test hydrogen sulfide production by growing it on HE agar plates. I think I've got a picture of that. Oh, here it is. HE agar plates, if the bacteria do not produce hydrogen sulfide, the colonies will not be black on the HE plate. If, on the other hand, the colonies are black, then the bacteria is producing hydrogen sulfide. Any questions about any of that? Here is triple sugar iron, whereas you can see it's black in the lower portion of the tube. It is not black in the portion of the tube which is growing aerobically. Any questions about hydrogen sulfide production? All right, let's talk about the triple sugar iron agar slant, or the TSI. The TSI tube has glucose, lactose, and sucrose in it. And then when we inoculate it, we're looking to see the fermentation of the sugar, both aerobically up here and anaerobically down here. The tube also has phenol red as its pH indicator in it, and the glucose, the lactose, and the sucrose are not present at equal concentration. The lactose and the sucrose are present 10 times higher than the glucose. There's also sodium thiosulfate and ferrous sulfate in the TSI, and these reagents are there to determine the hydrogen sulfide, if that is made by the tube, in which case it'll be black if hydrogen sulfide is made. All right, an inoculating needle is made to stab the deep. That's this portion of the tube. And you should go 2/3 of the way down. There are just a little over a half. And you mark it from the bottom of the slant there. And then you pull that out and then re-inoculate the needle with bacteria. And then you grow the bacteria aerobically on the slant. And then you look at the growth and whether fermentation happened. You can have many different results, and this table is going to show you the possibilities. I think this is all of the possibilities, but there could be something I was missing. It's most of the possibilities. You can have negative, where the sugar is not fermented, the hydrogen sulfide is not produced, no gas is produced in fermentation of the sugars. And remember, there's 3 different sugars: glucose, sucrose and lactose. You look both aerobically and anaerobically, and the sugars are not fermented in both. Now you look at tube 2, and you see that it's black, so we know that hydrogen sulfide is being produced. Because it is black, I also know that a sugar, at least one sugar, was fermented, meaning you only have hydrogen sulfide being produced when at least one sugar is fermented. You do not have hydrogen sulfide being produced when no sugars are fermented. So you have to have a fermentation reaction occurring when hydrogen sulfide is being produced. In this tube, it's black on the bottom, and because it's black, I know it's yellow. You can't see any yellow, but if we were to look at this earlier, we probably would have seen yellow, like on the top and on the sides, and then the black began on the bottom and then expanded. Okay, so fermentation was happening, and at the moment we don't know what, but we notice the slant is red. So fermentation looks like it's not happening on the slant. In reality, what's happening with tube 2 is glucose was fermented, and in the slant, all the glucose was used up because there's very little glucose. It's only 0.1% or 10 times less than lactose and sucrose. So only the glucose was fermented. And the reason why it's red is once all the glucose is used up, being fermented, the bacteria will then utilize proteins, and there is protein in the media. And when the bacteria metabolize proteins, ammonia is going to be made, which can be made into ammonium, and that both of those are alkaline, and so that will revert the color yellow to the color red because of the pH switch, meaning initially the color was yellow from the production of acids, but once the sugar is used up, because glucose is at low concentration, the bacteria will start digesting proteins, which will make alkaline products, and that will change the color from yellow to red. Any question about any of that? So we know only glucose is fermented because there is a sugar being fermented in the bottom, but not on the slant. And the reason is because it's glucose that was fermented, and all the glucose on the slant was used up. Why the lowercase can remain yellow when the slant turns reddish or pinkish is because the sugar will be used slower anaerobically than it will be aerobically, meaning the bacteria grow slower anaerobically, and it metabolizes the sugar, glucose, slower anaerobically than it does aerobically. Any questions about any of that? All right, tube 3 is similar to tube 2, except we don't have any hydrogen sulfide being produced, so it's not black on the bottom. We do have acid being produced on the bottom, but not on the top. So once again, we see that tube 3 has glucose being fermented, but not lactose or sucrose. And that's why the slant is reddish. Initially, it was yellow, but with time, all the sugar got used up. Then the proteins were being digested. The proteins made ammonia, which would make ammonium, and that would be basic, and that changed the color from yellow to red. Any question about any of that? All right, question 4 we see is yellow both anaerobically and aerobically. And therefore, we conclude the glucose is fermented in at least one of the other sugars. We don't know which. It could be lactose or sucrose. But because the slant is yellow, then either sucrose or lactose is being fermented. And the same in the bottom portion. You'll note that there's a gas bubble here and a break there and a break there, or a bubble there. And that's from gas being produced with the fermentation. And so we can see this one is acid being produced and then gas being produced as well. Any questions about TSI? You'll note that TSI is pretty difficult to interpret. I won't ask you anything hard, but I do ask you questions like which tube had gas produced, which tube had acid being produced. You have to read that as yellow. So yellow acid would be produced here, here, and here, as well as there and there initially. All right, that's it for the TSI test. Let's go on to the sulfide indole motility, or the SIMS auger deep test. What you do with this is that we have auger in a deep, and then we stab inoculate it with the needle, which we can actually see right here. Let me blow that up. Right there, you can see the stab mark on that one, and pretty much on that one, but that one's a little blurry. You can see it kind of on that one and very well on that one. We can't see it here because it's so dark, and the control probably didn't have any growth occurring. Anyways, you stab inoculate. Probably the first thing you should do is add Kovacs reagent to the tubes. And if Kovacs reagent remains yellow in color, like tube control and tube 5, then indole was not produced. If it turns pinkish or reddish on the top, because this floats, then indole was produced, and we can say that in SIMS tube that it is indole positive. So tube 1, 2, 3, and 4 are indole positive. Hydrogen sulfide was produced here because it's black in the lower portion. No hydrogen sulfide in any of these tubes, and of course not in the control. And then we look at the tube for motility. And what we're doing here is we're looking to see if the bacteria remain in the stab mark or if it comes out of the stab mark. Tube 3, it's not very clear, but there's no bacteria outside of the stab mark. Tube 5 as well, no bacteria outside of the stab mark. So those are not motile. Let me see if I got that right. 2 motile negative, and I think 5 motile negative. Motile negative. Tube 4 is clearly motile because the bacteria are in this region here and all of this region here. So the bacteria begun swimming out of the stab mark. I think tube 3 is non-motile. It's not terribly clear, but it's strongest there. And I don't think that haze is bacteria. I think that's just haze on the tube. So tube 2, 3, and 5 are all non-motile. Checking to see if I'm correct here. Motile negative for that one. Let's make sure 3 is. Motile negative. And tube 5, motile negative. Okay, I got it. Tube 4 and tube 3 are motile positive. That one isn't totally clear to me. but that one is. Did I read that wrong? Oh, no, I didn't. Motile negative, number 3. Motile positive, number 4. So I did read it right. And then hydrogen sulfide being produced in tube 1 and none of the other tubes. Any questions about how you read the SIMS tube? Like I said, you get 3 different tests out of the SIMS tube. Indole test, hydrogen sulfide production, and then motility. All right, let's talk briefly about the McConkey's auger plates called the MAC plates and the HE auger plates or the hectone enteric auger. The purpose in MAC and HE is to grow gram-negative enterics. So if the bacteria grows on these 2 plates, then it's likely to be a gram-negative bacteria. Gram-positive cells are selected against, and they usually will not grow on McConkey's and HE plates. For McConkey's, we're looking to see if it's a gram-negative, in which case it'll grow. We're also looking for lactose fermentation. If the bacteria ferment lactose, it'll be pinkish or purplish in color. And not only the colonies, but the area around it are pinkish or purplish. If it does not ferment the sugar lactose, it will be its normal color, in this case, off-white or white. Any questions about the MAC? All right, on the HE plate, if it ferments the sugar glucose, it'll be salmon in color, the colonies and the bacteria. If it does not ferment the sugar, lactose, it'll be not salmon in color. It'll most likely be the color of the bacteria, which most of the time will be white or off-white. Any questions about McConkey's and HE auger plates? All right, so in lab 9, you're not physically going to be doing any of the biochemical tests, but you are responsible for knowing how to perform and interpret these tests. You can view the following clips on carbohydrate fermentation, the SIMS test, the MRVP test, the triple sugar iron test, and the McConkey's auger. Also, view some of the website Microbiology. Click on differential tests to the left to expand the menu. Let me open that and show you. You only need to click on the test that you need to complete the table for number 1. So you'll need this information here in Microbiology to get information on the tests that we're running. For the worksheet to lab 9. And I'll show you that in a minute. It's under 2. Oh, we've got case studies here too. Up the test is on the worksheet. So let me go to the worksheet. So you have to fill in the worksheet to lab 9. In 1, you have to describe the oxidase test. Did you guys already do this? Maybe you did something similar to this. Anyways, describe the oxidase test and give the positive result and the negative result. I've got the positive result filled in for you to show you how to do this table. For the indole test, do the same, fill in the results, what color it is if it's positive, what color it is if it's negative. Do that for the MR test, the VP test, and the citrate test. Now, all of these you can get from this lab. For the carbohydrate fermentation, you can do this from the PR test, which is also discussed in this lab. An example, glucose. Give me the positive result, what color the tube is, if it's positive, and then the negative result. With the positive result here, you can either take that as acid or acid and gas, because in this test, there's actually more than one, well, more than two possibilities, we'll call it that way. And there's more than one positive. Acid is positive and acid and gas are positive. And you only need to discuss one or both, either way. For this test. And then tell me the positive result if gas is produced and the negative result if gas is produced. For TSI, the triple sugar iron, tell me what a positive result is and what a negative result is. Now for TSI, remember you got to give two results. You have to give the slant and you have to give the butt. And it's traditional that you give the information in the slant first or on top of the information in the butt. And it will differ. Let me go to that. It will differ depending on the test. For example, the slant here is a different color than the slant here is a different color than the butt. And here the slant is a different color than the butt also. All right. So TSI, then tell me what happens with the fermentation of lactose and or sucrose. And you have to give the results for the slant and the butt. And then a negative result for the slant and the butt. Hydrogen sulfide production. Give me what a positive result looks like and give me what a negative result looks like. For gas production in the TSI tube, you got the air pocket in the Durham inverted tube, no air pocket in the inverted Durham tube. For the Sims Auger Deep, sulfide indole motility, tell me what it looks like if sulfide is produced positive, and tell me what it looks like if sulfide is not produced. And then in the Sims Auger Deep, tell me what the tube looks like if it does produce indole, positive result, and then negative result for indole production. And then tell me what it looks like if the bacteria are motile, positive. Tell me what it looks like in the Sims tube if the bacteria are not motile. Any questions about that? And then lastly, tell me what it looks like in the MAC plate or both the MAC and the HE plates with lactose fermentation positive and then lactose fermentation negative. Any question about any of that? And to complete the table, it tries to give you a hint. What reagent must be added to detect the presence of indole? So name the reagent. B, methyl red is a pH indicator that remains a certain color. Tell me what the color is under acidic conditions. Anyone remember? All right. What test uses the Barrett's A and Barrett's B reagents to detect the presence of acetone, a neutral product formed from glucose metabolism? Anyone remember? Look it up if you don't. All right. Cinnamon citrate uses the pH indicator. Name the pH indicator. And it turns a blue color under either acidic or basic conditions. You can either bold your answer, or if you can't bold, just write your answer over here. So D does have two answers. You need both to answer D. What is the name of the pH indicator? And then is it acidic or a basic if the tube turns blue in color. 2E, what is the pH indicator used to detect carbohydrate fermentation? And is it yellow under acidic conditions or under basic conditions? Once again, bold your answer. If you can't bold, just write it over here. F, organisms that possess this structure test positive for motility. So name the cell structure. G, 3 organisms that produce hydrogen sulfide are, I can name 2, but look it up. It was actually mentioned in this lab. Although if you come up with another one, I will accept it. I'll just have to probably look it up because I don't remember all of the ones that produce hydrogen sulfide. H, if an organism ferments glucose but not lactose or sucrose, the TSI will be what color? In the deep, the slant will be what color? So that's the first one. And the deep will be what color? I, state the 3 tests for which SIM is used. So SIM stands for an abbreviation for something, and name the tests here. J, MAC and HE plates are both in that they inhibit the growth of gram-positive or gram-negative bacteria. So once again, you need to bold gram-positive or gram-negative. If you can't bold, just write it down here. And then MAC and HE plates are both something in that they inhibit the growth of gram-positive or gram-negative bacteria. So it's talking about a specific type of media there. And then 3, complete the case studies identified below. This gives you the key that you'll use for answering this question. Only use this key for this question because the tests and the species are different than our unknown project. So don't use this in the unknown project. In the following table, add the biochemical test results for each bacteria provided in the case studies. So case study 1, give me the results of the oxidase test. Give me the results of the glucose fermentation test. Give me the results of the lactose fermentation test. Give me the results of TSI. And this one, you might need more room to write the results. Tell me whether it's H2S producing or not. Tell me motility, is it modal or non-modal? Tell me, is it indole positive or indole negative? Tell me whether it's MR positive or MR negative. VP, tell me whether it is VP positive or VP negative. And then lastly, give me the results for a citrate. So put your results of the case study here, and to get these results, you got to look on the case studies. Let me see if I can go to that. No, that's not it. Case study 1. Before the results of the routine culture after overnight incubation in a CO2 incubator at 35 degrees C. So we streaked out our bacteria causing the infection, and then we streaked it out on a MAC plate. This is a blood agar plate, by the way. McConkey's plate, and then we did the gram stain shown here. This is rather light, but you should be able to see it. And then they did the catalase test. It was catalase positive. They did the oxidase test. It was oxidase negative. On day 2, we then ran these different tests. We did the SIMS test shown here. That's probably fermentation. Let me go back to the original case study. Wrong one. Yep, that's the wrong one too. All right, so these are the results. Write in your answers. And like I said, I'm trying to show you the results. So there's the SIMS tube A. Sorry, this got split. This is the MR test. That's probably the VP test. This is the citrate test. And I'm thinking that's triple sugar iron, but let me make sure. Yeah, triple sugar iron test. All right, any questions about what you're doing?
I have a question. In the first table under the triple sugar iron one, the results are either AA or KA. Can you remind me what that means?
Yeah, if I can get to it.
It's just up from there.
Yeah, I know.
I saw it for a second.
There it is. I told you that well, KK
Oh,
means
right.
it's alkaline in the butt and is alkaline in the slant. And let me see if I remember
Okay.
how you do this. You put the slant on top or first and then the butt. And so tube 2 is K in the slant, meaning it appears there's no fermentation in the slant, and there's fermentation or A acid, meaning it's yellow in the butt. You can't see it because of the black. And remember that in the slant here, initially it was yellow, but the glucose was all used up and then it became K or pinkish in color. Okay.
Okay. Yes. Thank you.
Okay, you got it?
Yeah, that made a lot more sense once I remembered that part.
Okay.
Thank you.
Sure, no problem. All right, then read through case study 2. And then these are the tests that are run on case study 2. Let me go to the worksheet. Put in your results. Gosh, I didn't see very many tests for 2. I must have missed something. Oh, here it is, day 2. Just need to go further. The SIMS test right here, MR test, the VP test, the Simmons citrate test or the citrate test, and then the triple sugar iron test. So write your results in the worksheet, table right here, and then figure out which bacteria is causing the infection in case study number 1, number 2, number 3, number 4, number 5. And use this key to figure out which bacteria is using it. For example, if the oxidase test is positive, there's only one that's positive, so you know it's Pseudomonas, which is causing that case study, if ever it's oxidase positive. If it's oxidase negative, then you got to look at different tests for trying to figure out which bacteria species cause the infection. Any questions about the case studies? All right, if not, go ahead and work on the case studies. I will be here at least until 8 o'clock or until the last student logs out, in which case I will log out as well. All right, so go ahead and get started. I'm going to stop sharing my screen now and stop recording as well.